very well that no preparation is complete unless you test yourself. And please do test yourself using some of the previous year's board questions itself so that you get an idea where you stand in your upcoming examination, maybe in 90 days or less than that. All right. So looking at all the previous year questions from this chapter, we will do it topic wise. First topic is DNA extraction, very simple topic wherein the question is mostly about enzymes that are used. So first question is about why protease is used. Very simple, to remove proteins. Second question, name the enzymes that are used during isolation of DNA from bacteria and fungi. Here, what is the difference between them? Their cell wall material is different. So I hope you remember from the video that we discussed. Anytime you start isolating the DNA, you need to get rid of the cell wall and everything else except for the DNA. So when we start with the cell wall of bacteria is made up of peptidoglycan, fungi is made up of chitin. So for bacterial cell wall, we need to use lysozyme, and fungus, we need to use chitinase. Subsequently, you would be using ribonucleases to remove RNA, and proteases to remove proteins. Going back to the next topic, that is restriction enzymes, I urge you to watch the video that I have made in which I have elaborately explained the mode of action of restriction enzymes, differences between endo and exonucleases, how to name a restriction enzyme, what is meant by sticky end, why are they formed, what are palindromic sequences, and much more. Quickly, let us answer all these questions as well. Um, Again, please do watch the video so that you get an idea in depth. I'm going too quickly, I understand. However, detailed videos are available in the same channel. How does EcoR1 specifically act on DNA molecule? How does it act? It looks for palindromic sequences. In case of EcoR1, what is the palindromic sequence you should know? Basis of naming restriction enzyme. How do you name it? First letter from the genus, two letters from the species, followed by the one letter from the strain that you use. What is EcoR1? How is it different from exonucleus? The same question in another way is, what is the difference between endo and exonucleases? What is the difference? One cuts from within, one from outside. Everything again explained in detail. How are sticky ends formed? So, how are sticky ends formed? They are formed because restriction enzymes always look for palindromic sequences on the double stranded DNA. What is that? It means that the same sequence present on opposite strands in the opposite order. Right? So when they cut it slightly away from the center, they would create some protruding ends or overhanging single-stranded portions we call as sticky ends. Why do we call them as sticky? Why do we call them as sticky? Because they have some hydrogen bonds and they have the ability to bind to the complementary cut counterparts of DNA. How does restriction enzyme function? Same as the previous question. Here they have asked about in general restriction enzyme. Here the first question, instead they have asked specifically about one of the restriction enzymes called as EcoR1. Again, same type of questions, palindromic nucleotide, what is meant by that, and the question you need to explain it with the diagram given in the textbook as well, children. Only then you get marks for that. Okay, then any two restriction enzymes other than SAL1, you can write any one that you have learned when you learn the structure of PBR 322. It could be HIND3, it could be EcoR1, any. You can even name PVU1 and so on. Describe the formation of rDNA by the action of EcoR1. Again, same picture that you are learning, same diagram that you're drawing for how EcoR1 cuts the vector and the foreign DNA, which is clearly mentioned in the textbook. And for explanation, please refer back to the video that I've already made. Explain the significance of palindromic nucleotide. Same thing, only that is the recognition sequence for the EcoR1, which can cut the vector as well as the foreign DNA. So that is the same question repeated in different forms if you notice. Again, I have just explained everything in the video. Alright, coming to gel electrophoresis, important questions. They can ask you to write the technique itself. Alright, so explaining the technique I have explained in the video. Please go through. Other questions is in another way the same question is asked. Suggest a technique a researcher who needs to separate fragments of DNA can use. So same technique, gel electrophoresis, explain in detail. Name the matrix used. I hope you remember this answer. Agarose gel. Where is it extracted from? If they ask, you should know. Seaweeds. What is the role of it? What is the role of it? It allows DNA to sieve through it. How does DNA fragments move on this matrix? What is the principle behind it? DNA is negatively charged, hence it would always try to move towards the positive terminal. Explain the basis on which gel electrophoresis works. Same thing. Okay. Explain in detail though. Mention how separated fragments can be visualized. ETBR. I hope you remember. Ethidium bromide and then UV light. You will see orange bands. PCR. Directly they can ask you the entire steps or they can ask you about the enzyme which is of high significance. Okay. The name of the enzyme is TAC from the bacterium Thermus aquaticus. Take T from here and AQ from aquaticus. One letter from the genus and two letters from the species. You get the name of the enzyme TAC polymerase. Why is it so special? It is special because it can withstand high temperatures. Again, same question, number 1 and 3 are the same. Source of thermostable DNA polymerase. Bacterium Thermus aquaticus. When you're writing, please underline it. When we are typed it, we will always italicize it. Mention the source of it. Done. Why is TAC polymerase preferred in PCR? Due to its thermostability. As you remember, PCR 3 steps. First step is to do with heat, denaturation, then binding of primer, helping in annealing, then extension. Right? So for that high temperature, only this TAC polymerase can withstand. Mention the role of primer, high temperature and TAC polymerase. Self-explanatory in the video. Still quickly, let's go and understand it. Primer are tiny sequences of small sequences of nucleotides which can go and bind to the separated strands of DNA, one of it, and then it can result in the DNA polymerase to extend it. High temperature for denaturation step. TAC polymerase is the our DNA polymerase that helps in polymerization. Okay, PCR applicatory type questions. Please read. Causative agents of HIV, AIDS, and COVID-19 belong to the same group of viruses. To diagnose and amplify the genetic material for further study of COVID-19 virus, RT-PCR technique is carried out. What do you understand by RT-PCR? What are the steps of PCR technique? RT stands for reverse transcriptase, which is probably not something that you have learned from the textbook. However, reverse transcriptase. The steps of PCR technique you know. So please write all the 3 steps, get the diagram, fetch the 3 marks or the 5 marks that it can get. A cell-free method of amplifying DNA first was developed in 1980s, which revolutionized the field of biotechnology. Name the method and explain the basic steps. Same thing, it is PCR, explain the steps. The next set of questions is about the topic recombination. Before we go into the questions, things to remember. The first recombinant DNA was made by Cohen and Boyer, 1972. Here is important for you to remember. What did they isolate? They isolated an antibiotic resistance gene from a plasmid of Salmonella typhimurium bacterium. And then what did they do? They isolated that antibiotic resistance gene from this organism and linked it with another plasmid, transferred it into E. coli, another bacteria which is similar to Salmonella typhimurium. That is what they did, year 1972. Alright, the questions from this. A host cell must be made competent before it is able to receive rDNA. Or how can we justify this statement? A DNA is negatively charged and it is very incompetent when it comes to getting inserted into a cell by itself. So what do you have to do? You have to change the host cell in such a way that it is able to take up the rDNA. or in other words, you are making this host cell competent. How can you do this? This can be done in stepwise procedure. First, you add calcium divalent ion, that is calcium 2 plus ions. Then on ice you're keeping both the host cell and the plasmid. After that, heat shock, back to ice. I'll repeat: calcium, ice, heat shock, ice. Clear? Okay. Then next question is the same thing. Another way it is asked. List any two ways of achieving this. You are just explaining the procedure. Why a host cell must be made competent before it is able to receive rRNA. Same thing. Role of calcium ions. It increases the permeability of the cell wall. Right. The steps that you would suggest to undertake to obtain a foreign gene product. For this, you are writing all the steps which are isolation of a gene of interest, extracting the DNA. After that, you are cutting it using restriction enzymes, putting it on a gel, isolating only that gene of interest. After that, amplifying it using PCR, inserting it into a host cell either in the direct method or vector-based method. Once you do that, story is not over. You have to check for transformants using selectable marker. Once you have that, what do? You go for either large scale production using bioreactors, or you do small scale production in the lab. Once you extract the product and then you take it out of the bioreactor, you cannot directly use it. You have to refine it, and the process of refining is called as downstream processing. So all the steps you have to write for this question, steps that you would suggest to undertake to obtain a foreign gene product. Next question is regarding, next set of questions is on the topic direct gene transfer. For which type of cell, which type of direct transfer of DNA is used? Gene gun, I hope you remember, only for plants. Microinjections only, microinjection is only for animals. How can retrovirus be used efficiently in biotechnology experiments in spite of them being disease causing? So what is the key word here? Disarm. Disarming the bacteriophage you had learned. Here you are writing disarming the retrovirus. The next question, non-viral and non-vector methods are used sometimes in gene transfer. What are they otherwise called as? They are otherwise called as the direct gene transfer methods, which are microinjection, gene gun, etc. Electrophoresis, and so on. Okay. So any two you can write. Selectable marker questions. Explain how to find whether an E. coli bacterium has transformed or not using a selectable marker. Answer. Okay. When a recombinant DNA bearing, yeah. So what can we use as selectable marker? In this case, the question is recombinant DNA bearing ampicillin resistant gene is transferred into a host. Now, how can you check if that host has got the plasmid or not? Plate it on a nutrient medium containing ampicillin. If the bacteria is having this gene, how from where? From the plasmid. If the host cell got this gene, that means it can be resistant to that antibiotic. Very clearly, I have explained the entire topic in the video. Please go through it. If you're not able to understand, I suggest you go through every video and then come back to this set of questions. How does antibiotic resistant gene act as a selectable marker? On this, I have made an entire video. In simple words, antibiotic resistance gene select the transformants on an antibiotic medium. Simple. But explain it. Listen to the video. Explain it with one example. You can take example as, let us say, ampicillin resistance gene. How do you use that as a selectable marker? If the host cell is able to grow on a medium containing ampicillin antibiotic. You know, antibiotic is something that kills any bacteria. If this particular bacteria is able to grow on an antibiotic called ampicillin, it should definitely have that gene called as antibiotic resistance gene. Where did it get? It was inserted using our RDNA technology, right? So um, for using it as a marker, how does it work? Please explain it in detail listening to the video. How does beta-galactosidase coding sequence act as a selectable marker? I don't think I have explained this in the video. Let me explain in simple words. Beta-galactosidase gene gives a particular blue color on a medium. Alright, what does it do? Beta-galactosidase gene will produce the beta-galactosidase enzyme. This is a gene. Gene will produce a protein. In this case, the protein's name is beta-galactosidase enzyme. It is an enzyme. This protein is a particular enzyme. Clear? And it gives blue color. Okay, so it gives blue color normally. Now, how is this used as a selectable marker is our question. So where is this found right now? It is found on, where is this beta-galactosidase gene found? It is found on our plasmid. Alright, so where are we inserting our gene of interest? We are inserting our gene of interest into this coding sequence. When I insert anything into a already existing sequence, the sequence is disrupted. I hope you understand that much. Yes, that means the plasmid is no more going to make beta-galactosidase enzyme. So therefore, no beta-galactosidase enzyme, no blue color. So if on the plate a particular colony is not having blue, producing blue color, it shows that it is a transformant. It is somewhat clear. If not, please go back and watch the selectable marker video. After you watch that, this will become very clear. Next question about insertional inactivation. Let me go back and tell you one thing. This beta-galactosidase coding sequence, we interrupted using our gene of interest, right? That is itself is called as insertional inactivation. All right. Insertionally, we inactivated one particular gene. In that case, it was beta-galactosidase. All right. Next possible question from this is they can directly ask what is insertional inactivation, give an example, or they can ask questions like this. State what happens when an alien gene is ligated at PVU1 site of PBR322, SAL1 site of PBR322. For that, I wanted to know one thing, that is, you should be able to draw PBR322 structure by yourself. Please remember, it has only 4 genes, ORI, ROP, TET, R and AMP-R. Every other arrow that you see are sites for restriction enzymes like SAL1, BAMH1. These are the restriction enzyme sites. These are the names of the restriction enzyme. The arrow goes to show where that can cut. Now, going back to our question, with this question, they are not going to give you this diagram. I just put it up so that you can revise what is it that you have to learn. So, for answering this question, you should know the structure of PBR322, which you should be learning either way, because this can in itself be a 2-mark question. Okay, so now our question is PVU1 site. Where is PVU1 site? Here. What does the question say? An alien gene, a new gene or a gene of interest, we have ligated into this part, right? We have been inserted here. What happened? What happened to the plasmid? That is a question. What will happen to the plasmid is we are cutting through one gene. Its name is AMP resistance. Therefore, this gene will be insertionally inactivated. The host cell will not be any more resistant to ampicillin. The same way, other question, SAL site. Here, this cuts the tetracycline gene, tetracycline resistance gene. Therefore, the resistant gene is cut. Therefore, no more resistance to its antibiotic tetracycline. Therefore, the host cell will not be able to grow on tetracycline. Simple questions coming from the topic bioreactor. direct question. What are they? Name the most commonly used. If they ask, is this simple? Most commonly asked is simple stirred tank. Two commonly used, simple as well as sparged stirred tank. How does it work? All the explanation, like it's a cylindrical tank, provides optimal conditions of pH, temperature, exponential growth phase, all those points that I explained already in the video needs to be included. Same question again in different ways is asked. How does development of this help large scale production? Okay. And next question is need label diagram of simple state tank. That could also come. So all the best for your exam.